Welcome back to IGCSE Biology Enzymes topic. This is the lecture number 3. In the first two videos, we covered what enzymes are, how they work, and how temperature and pH affect them. This video is different and very important. This is about practical skills. Cambridge IGCSE has a separate paper about it. Paper number 6. That test your ability to plan, carry out, and evaluate experiments on enzymes. Here is your exam question. Read it carefully. A student investigates the effect of temperature on enzymes activity. State 3 variables that must be kept constant. This is a classic paper 6 question. By the end of this video, you will answer this easily and understand every type of practical question Cambridge can ask about enzymes. Let's go. Before we look at the actual experiments, you need to understand variables. There are three different types of variables. First one is the independent variable. This is the one thing you deliberately change in the experiment. Like if you are testing the effect of temperature on enzymes, then temperature is the thing you are changing and setting its different values like 10, 20, 30, 40 or 50 degrees and see what's happening. So this temperature is your independent variable. This second is dependent variable. This is the thing what you measure, what changes as a result of your independent variable. In enzyme experiments, this is usually the rate of reaction measured by how quickly a color changes or how long does it take for starch to disappear. So basically, the changes in the reaction, the changes what you observe as a result of your independent variable is your dependent variable. The third is controlled variables. These are everything else that must stay same to make it a fair test. Like if you change temperature but also accidentally change pH, then you cannot be sure which one caused the result. So pH, enzyme concentration, substrate concentration, volume, all must stay constant. You must note these things down as the examiner will ask you to identify them in almost every practical question. Now let's look at the first classic IGCSE enzyme experiment. Catalase is found in almost all living cells and we are testing how temperature affects catalase activity. Catalase is found in celery, in potato. Its job is to break down hydrogen peroxide in water and oxygen. When it works, oxygen bubbles are produced. You can actually see it happening. There are 4 steps of experiment you have to follow with me. Step 1 is take celery or potato, blend it with water, then filter it. Now you have a catalase solution. Step 2. Take small filter paper discs and soak them in the catalase solution for 1 minute. The disc absorbs the catalase. Step 3. Set up 5 different beakers with hydrogen peroxide solution. Place each beaker in a water bath at a different temperature, like that is 10 degree, 20 degree, 30 degree, 40 degree or 50 degrees Celsius. So temperature is our independent variable, the only and one thing we are changing. Step 4 is drop the catalase discs into hydrogen peroxide. Catalase breaks down the hydrogen peroxide, oxygen bubbles will form. Bubbles will be collected on the disc and the disc rises to the surface of beaker. Now time, how long the disc takes to rise. Shorter time means fastest reaction and the higher enzyme activity. Most importantly, you have to repeat 3 times at each temperature and calculate the mean value. This reduces anomalous results and increases reliability. After that, you can plot your graph by taking temperature on x-axis and time on y-axis. Now the second experiment is testing the effect of pH on amylase activity. First, you have to remember a keyword: buffer solution. A buffer solution is a liquid that has a known fixed pH. It keeps that pH constant throughout the experiment even if chemical reactions are taking place. You must use buffer solutions when testing pH, otherwise the pH might change during the experiment and your results would not be valid. Here is your method. You prepare starch solution and amylase solution. You have to set up 5 different test tubes, each with a different buffer solution, like for example pH 4, pH 5, pH 6, pH 7 and pH 8. Add the same volume of starch to each tube and same volume of amylase to each tube. Now, after every 30 seconds, take a small sample from each tube and add a drop of iodine solution into it. If starch is still present, the iodine turns blue-black. When the starch is completely broken down, the iodine stays brown. Record the time taken for starch to completely disappear at each pH. The shortest time means the fastest reaction. It means the shortest time that will be taken for starch to completely break down and disappear means the enzyme worked best at that pH, and that is the optimum pH value. Your control variables are temperature, volume of starch, concentration of amylase, as they all must stay constant. Now, the pH graph that you must understand. On x-axis shows pH from 1 to 10, y-axis shows rate of reaction. Two curves on this graph. The red curve is pepsin, the enzyme in your stomach. Its optimum pH is 2, very acidic. At pH 2 the rate is highest. If go above or below pH 2, rate drops sharply. Active site changes its shape and an enzyme gets denatured. The green curve represents most enzymes in our body which works around 7 pH, neutral pH. So at pH 7, their rate of reaction is highest. If we go too far above or below that pH value, then enzyme can get denatured. Now look at the right side, how to describe this graph. Below the optimum, rate is low, active site shape is not ideal for the substrate. At the optimum, rate is highest. Active site shape perfectly suits the substrate. Above the optimum, rate falls. Active site changes shape and denaturation. Write this down that a pH well above or below the optimum causes the enzyme to denature. And the most important point is different enzymes have different optimum pH values. Like pepsin works in acid because your stomach is acidic. Most enzymes work at pH 7 because that is the environment inside body cells. So different enzymes, different optimum pH values. Now this is the examiner trap time. 3 mistakes on practical questions that cost marks every year. Trap 1. Keep everything the same is not enough. I see this constantly. Students write keep all other variables same and get 0 marks. Why? Because the examiner wants you to name the controlled variables specifically. Write it down as keep pH constant, keep enzyme concentration constant, keep substrate concentration constant, keep volume constant. Specific names are required. Trap 2. Forgetting to repeat and calculate the mean. Never just say record the time. Say repeat 3 times at each temperature and calculate the mean time to reduce the effect of anomalous results and increase reliability of experiment. Trap number 3 is confusing independent and dependent variables. If you are testing the effect of temperature, temperature is what you change. So this is independent variable. And the rate of reaction is what you measure. So this is the dependent variable. These are mixed up in student answers constantly. Know these three traps and you will collect marks that most students drop. Do you remember this question from the start? Here it is again. A student investigates the effect of temperature on enzyme activity. State three variables that must be kept constant. Here is your answer. 3 marks, 3 points. 1. pH of the solution. 2. Concentration of the enzyme. 3. Concentration or volume of substrate. These 3 will always get you full marks. Look at the box below. There are other acceptable answers too, like volume of solution, size of filter paper disc, same source of enzyme. Any 3 specific named variables will score marks. The key word is specific. Name them. Do not just say everything else. Name them individually. You must log three things into your mind before your exam. Number 1. Name your variables specifically. Independent is what you change. Dependent is what you measure. Controlled variables must be named individually. Never write everything else stays the same. Name them. Each one can be a separate mark. Number 2. Always repeat 3 times and calculate the mean. And say why. To reduce the effect of anomalous results and increase reliability. These exact words are in the Cambridge mark scheme. Number 3. Buffer solution means fixed pH. When any experiment tests the effect of pH on enzymes, buffer solutions are used to keep pH constant. Know this term, know why it is used, and you will not be caught off guard. That is video 3. Chapter 5 Enzymes practical skills are fully covered now. In the next and final video on enzymes, we are doing a complete past paper walkthrough. Every type of question Cambridge can ask about enzymes. Solve them step by step with mark scheme language. Subscribe so you do not miss it. See you in the next one.